My name's Elliot Solis, and I'm a scientific curator for the GWAS Catalog. And today I'm going to walk you through how to submit your own data to the GWAS Catalog. And this is an update of a previous webinar from 2021. So I'll be explaining a few changes that have been made to the process since then. I'm going to start with a brief overview of GWAS summary statistics and mention some of the benefits of sharing these datasets, both for the researchers that produce them and for the wider scientific community. I'm also going to talk about some of the reasons why the GWAS Catalog is a good place to submit your summary statistics. I'll talk about the eligibility criteria that we have and explain our standard summary statistics format, including some upcoming changes to the format. I'll then give a step-by-step guide to the submission process and finally explain what happens to your summary statistics after submission and how you and others can access them. So I won't have time to cover every possible scenario today. So if you are ready to submit your own data, I'd encourage you to also look at our detailed submission instructions on our website, as well as our FAQ page. And if you're interested in more general information about the GWAS Catalog, including how to access the data in the catalog, please check out the related resources page on our website, which also has links to some of our other training materials. So to start with the introduction, summary statistics, we mean by that the full set of association data for all of the variants that were analyzed in a single GWAS. So basically the same data that would go into producing a single Manhattan plot like this. The summary statistics usually take the form of a table of variance and p-values with one row for every variant. And they usually have a lot of other information, such as the effect size, shown here as a beta. And importantly, full summary statistics contain a lot more data than is usually reported in journal articles, which would tend to just focus on the most significant associations. So summary statistics can be valuable for a number of different purposes, including GWAS meta-analyses, calculating polygenic risk scores, and Mendelian randomization studies, and also to analyze genetic correlations between different traits, to estimate SNP heritability, or to integrate GWAS with other omics datasets. By sharing your summary statistics, you as the producer can also benefit at the most basic level by fulfilling the submission requirements of a journal, but also it can greatly increase the citations that you get for your work because others are reusing your data and therefore citing it. Sharing the summary statistics can also improve the reproducibility of your work, and that can build trust as well in the results. And sharing your own data can also encourage others to do the same, so contributing to a growing collection of publicly available summary statistics that you can also make use of. Now introducing the GWAS Catalog's role. The GWAS Catalog is a comprehensive resource of variant trait associations from human GWAS. The catalog contains two main types of data. So we have top associations from published GWAS, and those are manually curated from papers by our curation team. But we also include the full summary statistics from both published and pre-publication GWAS. And these can be submitted directly to us by authors. The catalog is a good place to submit your summary statistics because it's widely recognized in the field and highly accessed. We also enable submission either before or after publication, and even in cases where there's no intention to publish the work in a journal. Our specialist curators are also there to support you with the submission process if needed. And we also have a number of features in place to make the summary statistics collection fair. So that means findable, accessible, interoperable, and reusable. These include providing unique and stable identifiers for each set of summary statistics, giving them rich annotations and supporting information, and packaging all of them together into a searchable resource. It's free to access and publicly available through a web interface via FTP and an API. And we enable interoperability across datasets by requiring a consistent standard summary statistics format for all submissions. And we also create harmonized versions of the summary statistics files that we receive to align them all to the same genome assembly and strand, and by providing structured annotations to enable comparisons between datasets. And finally, we have a clear standard usage license, and we collect detailed provenance information about all of the summary statistics. And our license doesn't place any restrictions on the reuse of the data. So the eligibility criteria for submitting your data to the GWAS catalog are that they must be for a human GWAS. We accept data for both genome-wide and targeted or exome-based array, exome-based association studies, and those can include array-based and sequencing-based studies. But in all cases, we need at least 100,000 variants to have been analyzed in the study. The summary statistics files must also conform to our standard format. So that means that there should be a separate file for each GWAS rather than being all grouped together. So that's normally one set of p-values in each file. And it has to be a tab-separated TSV file, and it must conform to our summary statistics standard, which I'll talk about now. So as I mentioned, we will be introducing a new and improved standard format at the end of March this year, 2023. And the main changes to the format from the previous one are that we've added some additional mandatory fields that were requested by the wider GWAS community as really important to have. And these also now have to be presented in a specific order to keep the files as consistent as possible. We've also got a few additional recommended fields that can help to increase the usability of the data set. And we've updated some of the metadata requirements to describe the study as a whole. You can find further information about the new format and how it was developed in this article, which is on bioRxiv. But I'll talk about the main points now. So for a successful submission, the summary statistics file has to contain the following 8 columns in this specific order, using the specified column headers. So they're numbered from column 0 to 7. So the first one is the chromosome number, followed by the base pair location, and then the effect allele, so the allele that's associated with the effect that you're reporting, and then the other allele, or non-effect allele. And then in column 4, you can enter the effect size, and you can choose whether you want to represent that as a beta, an odds ratio, or a hazard ratio, depending on how your study works. And you should then use the appropriate column header, depending on which one it is. A quick note here that if you want to provide the effect size in more than one form, so maybe as a beta and as an odds ratio, you can only put one of them in that column position 4, but you can add the others at the end of the file as an optional additional column. So the column 5 is the standard error of the effect, followed by the frequency of the effect allele in your sample, and column 7 is the p-value. So after that, there is a set of encouraged columns. So these are standardized, but they're not mandatory. But we do highly recommend that you include them to make your file as rich and informative as possible. And for these, you also need to use the standard column headers that are listed here. So these are the upper confidence interval of the effect, the lower confidence interval, the RSID of the variant, if it has one, and then another column called variant ID, which is where you can give an internal variant identifier, which is just formed by concatenating the chromosome, the base pair location, and then the ref and alt allele. Next is the info or imputation information metric. N is the sample size for this variant, and ref allele is a column to indicate which of the 2 alleles that you reported, the effect and the other, which of those is the reference. And there are more detailed information, instructions about specific column requirements in our documentation on the website. Finally, you can include any number of additional columns that are important for your results. These won't have standardized column names, so instead we ask you to provide an explanatory legend along with your submission, which I'll go over a little bit later. There are a few important considerations that I want to point out for the summary statistics format. So first, for the effect allele and other allele, you can only enter nucleotide symbols here, so A, T, C, or G. And that applies also to insertions and deletions. So they should be written out in full in terms of the nucleotides that are present or absent in each allele. We don't accept symbols like I and D. We also ask for a separate column. We don't ask for separate columns to provide the reference and alternative allele. But in this ref allele column, you can state whether the reference allele is the effect allele, if it's the other allele, or if it's unknown in this case. And you can also provide the ref and alt alleles as part of that variant ID string. For the base pair location, it's important to know which coordinate system has been used in your file. So this can be either zero-based, where the first base of the chromosome is called position 1, or it can be zero-based, where the first base of the chromosome is position 0. And you'll be able to state which of these was used in the metadata file, which I'll show you later. P-values should be represented in full so that they can be of most use to downstream users. So you should avoid rounding very small p-values to 0 or to an arbitrary cutoff wherever possible. If your analysis software just can't provide such small p-values with precision, you then should provide the information about the analysis software that you used along with your submission so that users can interpret those rounded p-values correctly. It's also possible to express the p-values as negative log values. And in both cases, whichever one you've done, you can provide information in the metadata later to explain it by indicating the analysis software and by saying whether or not negative log 10 p-values have been used. Finally, about the effect allele frequency. This is a mandatory column, but we do recognize that in extraordinary situations, there are sometimes privacy concerns related to providing allele frequencies for rare alleles. This is mainly the case in small cohorts with sensitive phenotypes. So where these concerns might otherwise be a barrier to sharing the data, you can specify a cutoff for the minor allele frequency, and anything below that, you can round up to mask the true values. If you do need to do this, you must provide the minor allele frequency lower limit in the metadata. However, masking allele frequencies does limit the downstream utility of the data. So wherever possible, we do encourage you to submit full allele frequencies. So to validate your files prior to submission, we provide a validator that can be run locally on your computer. So this ensures that you've prepared your files in the standard format before you get started with the submission process. And that can save time later on. So I've added a link to the full documentation here. And one thing to note is that this validator requires you to have Python version 3.9 or later. It's quite simple to run. If your file is valid, it will display a simple message saying validation status true. But then if there's an error in the file anywhere, it will show you the primary reason for the validation file. In this case, it's telling me that the main reason is that there's an issue with the headers. And in this case, the file has failed because one of the mandatory columns, chromosome, was not found in the expected place. So probably I just missed that file out when I was missed that column out when I was preparing the file. So in addition to your valid summary statistics, you'll need to have a certain amount of supporting information at hand as well. If you're submitting data for a published article that's already included in the GWAS catalog, so especially for past GWAS papers, then metadata has probably already been curated by our team. And in that case, you will only need to provide the summary statistics and some basic file information. On the other hand, for pre-publication submissions, or for just more recent publications, our curators may not have reviewed the study details yet. And so in those cases, we'll ask you to provide the summary statistics as well as more detailed study and sample metadata. I'm going to focus on the second scenario because there are a few more steps to tell you about. but a lot of this will also apply to the first scenario, which is just a bit simpler. So I'm going to use a fictional example to illustrate the process. So imagine that my lab has completed a GWAS project about pneumonia, and we've submitted a manuscript to the fictional Journal of Lung Diseases, and they've asked us to submit the summary statistics to a repository and also to get an accession number that's stable that we can include in the manuscript. So in this example, we might have 3 different studies in the publication with summary statistics files for each. So we could have a case control study looking at pneumonia, then a quantitative study looking at the severity of the disease in those patients. And we might have another case control study looking at chest pain among the pneumonia patients. And each study might contain 100,000 SNPs genotyped on Illumina and Affymetrix arrays. And each study could be a meta-analysis of 2 different cohorts. So we might have one of Chinese ancestry with a certain number of cases and controls, which were recruited in both China and Singapore. And that could be meta-analyzed with another cohort of European ancestry with a certain number of cases and controls recruited in the USA and Australia. And I'll use these details to illustrate some of the steps in the next section. So to submit, there are 5 steps in the process, which I'll go through one by one. And remember that if you need help, you can check out the documentation and our FAQs, or contact us on our GWAS-subs@ebi.ac.uk help desk email line. So the first step is to create an account. So when you go to our submission website, you'll be prompted, if you don't have one already, to create a LS login, which is administered by Life Science RI. And you can use this to log into our submission interface. So you just need to go to the link that I provided there and follow the steps. And you can use an existing login, such as your own institutional account or something like Orchid or Google to set up your ID, or you can start from scratch. And you then need to create a GWAS account. So GWAS is a tool that's used to transfer large files. So you can go to this URL and follow the installation instructions for your operating system to install GWAS Connect Personal on your computer. And during that installation process, you will also create a GWAS account. And it's easiest to use the same email that you used for your life science login. But either way, take note of the email address that you've associated with your GWAS Connect Personal, because you'll need that later. And you'll also create something called a GWAS endpoint, which is basically a connection between GWAS and your computer's file system. And you can give that a name that you'll remember. So the next step is to prepare your summary statistics files. You need to make sure that your summary statistics conform to our standard format from earlier. So in my example, I would make sure that I had 3 separate files for the 3 different studies. I would make sure that they were all in a consistent TSV format, which would look something like this. And make sure that the mandatory columns are all there and they're all in the correct order. And then make sure that any standard column headers that I've used, that any of the recommended columns that I've added have the standard column headers, for example, for RSID here. And it's always a good idea to double check using the offline validator to make sure the summary statistics are all valid before you get started with the submission. So once your summary statistics are validated, it's time to create a submission on our portal. You can start by going to the GWAS Catalog submission homepage and completing the questionnaire. This will guide you to the right submission form for your situation. So for the example that I showed earlier, I would just click on Get Started and then that would take us to these questions. So first it asks, are these GWAS described in a published manuscript? So in my example, we were just submitting it to a journal, so we would say no to this. Are these GWAS described in a manuscript submitted or accepted for publication? In this case, yes. And then this would give us a form that's customized to the questions that we'd answered already. And in this case, for a new accepted or submitted manuscript. And it first asks for a bit of detail about the work itself. So a title of the manuscript, and then a description, which you can write fresh here, or most normal thing to do would be to enter the abstract that you've used in the manuscript. It then asks for the journal name, and here you can click to select something from a drop-down list, or you can type it in. So for this fictional example, we'll just copy and paste the name of the fictional journal. And then it asks for some author information. So that starts with the first author, and the first author can be either an individual or a consortium. So in this case, we're going to go with an individual name and enter the first name or given name and any middle initials in the first field, and then last name or surname in the second field, and an email address to contact the first author. For the last author, we're going to enter a consortium as an example for this one. So again, that just goes in that section there, and with an email as well. And probably the more important thing here is the corresponding author. So this is the author that we as the GWAS Catalog will contact to update on the progress of the submission. And it's also the person that will be contacted if there are any queries about the data set from other users. And it's also where all of the information about the submission will be sent. So the email in that case is mandatory. And if the manuscript's already associated with a preprint, you can enter details about that here as well. So you can enter the server where the preprint is hosted, and also a DOI. And we'll use that to keep track of the provenance of the summary statistics files. And finally, there's a section where you can enter an embargo. So if you're happy for the data to be released to the public immediately, you can leave it with the default date, which will be the date of creation. So that's an old video, so it has an old date on it. You can also set a specific date if you want it to be released maybe in 2 weeks' time. And the final option that you can choose is to embargo until published. So that means that we won't release the data unless the paper has been published and has a PubMed ID, or if you tell us that you want to change it and release it early. So you then can click submit, and that will create the submission. And it'll give you this summary that shows all the details that you've entered at the top, as well as a submission checklist at the bottom. Here you need to confirm that you've completed all the things in the checklist. So creating your login, installing GWAS Connect Personal, and you need to also enter the email from your GWAS account in this box. If you are not quite sure which email is associated with your GWAS account, this link here can tell you which one it is. And we also ask submitters to agree to share their data under a CC0 Universal Public Domain Declaration. This is an unambiguous declaration of free and open access and allows downstream users to consume the data without restriction. Once you've agreed to all the items in the checklist, you can click Create Submission. You'll then see a page where you can start uploading files. And it's next time to upload your summary statistics. So first you can click Upload Summary Statistics, and that will open GWAS File Manager in a new tab in your browser. On the left of GWAS File Manager, you can see the GWAS Catalog folder where your summary statistics will end up. And on the right, you can click Search to find the files on your own computer. Click Your Collections, and then find the Globus Connect personal endpoint that you named earlier when you were setting it up. In my case, I've called it My Summary Statistics. You can then drag and drop your summary statistics from your own computer on the right to the GWAS Catalog folder on the left. And there's no need to wait on this page for the transfer to complete. You'll get an email to tell you when the transfer is finished. So finally, it's time to upload your metadata. Remember that if your studies are already in the GWAS Catalog, you'll only need to provide the summary statistics themselves. Whereas if it's pre-published or only recently published, we'll probably ask you to provide additional metadata as well. So first click Download Submission Form to get an Excel spreadsheet where you'll enter your metadata. The spreadsheet has two sheets, study and sample, and the required columns are highlighted. And there are some helpful tips and examples provided in the column descriptions. Some of the columns accept free text, while for others, you can only select from a limited set of options listed in the documentation. For a lot of the columns, it's possible to enter more than one value, separated by the pipe symbol. And this is indicated in the column description. I won't have time today to go through the requirements for every field in detail, but there are detailed instructions in our documentation that will explain each field. And I'll just give a summary and highlight a few of the specific ones today. So the study sheet is where you can enter information about the genotyping methods, the analysis and statistical methods, the traits that were under investigation in each study, and the properties of the summary statistics files themselves. You should include one row in this spreadsheet for each individual GWAS in your submission. So in my example from earlier, I should have 3 rows for each of the traits that I studied: pneumonia, pneumonia severity, and chest pain in pneumonia. The first column in the study sheet is the study tag. So this is a free text label that you can give to uniquely identify each study. And it will be used to match up the information between the studies and sample sheets. Next is the genotyping information, including the genotyping technology, whether it was an array or sequencing, for example, the array manufacturer, and any additional information you want to give about the array. Then there is some information about the analysis, including the software used for the association tests, imputation information, the number of variants analyzed, and you can also go into more detail about the statistical model and any covariates that were included. And you can also list any named cohorts that were used in the study and indicate if the study was sex stratified. Then there are 2 trait fields. So in my example from earlier, when we were looking for associations with chest pain in patients with pneumonia, in this case, chest pain we would call the reported trait. This is the main trait under investigation, and the variants listed in the summary statistics are being tested for association with this trait. Whereas pneumonia in this case would be the background trait. So a characteristic that all of the study participants share, but the variants in the summary statistics are not being tested for association with pneumonia in this case. It's rather just a part of the background context of the study. Oops. Finally, there's information about the summary statistics files that you've uploaded. So these include the file names, as well as an MD5 checksum that we use to make sure that we've received the same file that you sent us without any errors or corrupted files. This is a hexadecimal number that you can generate using a simple command line operation. And we provide instructions to calculate it using various operating systems on our website. I'll mention the Readme text field on the next slide. And then there is also information you can enter about the genome assembly used for your summary statistics. And then fields where you can state whether the p-values are in negative log 10 format or in normal scientific notation, whether you set a lower limit to mask small minor allele frequencies, and which coordinate system was used for the base pair location. In the Readme text column, we ask you to enter some author notes that will appear alongside your summary statistics files. These should be formatted as a numbered list, including the citation for your work in a standard style, a legend for any non-standard columns that you included in the summary statistics, and then any other useful comments, such as specific QC measures that were applied to your files or other information. Please copy-paste these author notes directly into the cell in this submission form in full. Please don't upload a separate README text file via GWAS, because the way things are set up, any additional files other than summary statistics files that you might transfer with GWAS won't be used. The sample sheet then contains information about the study participants, including the number of individuals, cases and controls, their ancestry, and the countries of recruitment. So in my example from earlier, I had 2 case control cohorts with Chinese and European ancestry, recruited across 4 different countries. In the study tab, you should create 1 new row for each GWAS, in this case, the 3 different pneumonia studies. And within each of those, you should also create a separate row for each broad ancestry category that was included in the study. So in this case, we had 2 broad categories: East Asian for the Chinese ancestry cohort and European. Altogether, that would give us 6 rows. Optionally, you can also create separate rows for replication cohorts, that is, samples that were not included in your summary statistics, but that were used for subsequent validation of the top associations, as well as separate rows for sex or age stratified groups that might be included, or for categorical or ordinal trait groups, such as mild, moderate, and severe cases. If you have a complex study design, please contact us for help. So the first column of the sample sheet is, again, the study tag. So you should make sure that you use the same study tags that you used in the study sheet so that they can be matched up correctly. Next is the general sample information, such as the number of individuals, cases, and controls. And this is followed by the broad ancestry category, which is a required field, and optional fields to go into more detail, providing free text for more granular ancestry descriptors, as well as the country of recruitment. The ancestry categories must be taken from the GWAS Catalog standard set of ancestry categories, which are described in more detail on our website. But this is the list here as well. So once you've finished entering the metadata in the submission form with the study and sample information all complete, you can click Submit Submission Form to upload the completed file and start validation. So the validation pipeline first checks the submission form, and if it fails, you'll see an error message to tell you what needs to be fixed. You can click Review Submission to download the failed form, and click Reset to remove it and start again. But if the submission form is okay, it will then go on to check the summary statistics themselves. If this step fails, you'll receive an error, and you can click Reset to edit your files and start again. This step can take some time if you have to make a few changes. So you probably want to run the offline validator to check your files before you upload them to GWAS and try again. Finally, if everything is valid, you'll receive an email confirming that your submission is successful. And this includes a link to the list of accession numbers for each of the submitted studies. So then your submission is complete. What happens after submission? So for submissions associated with a published article, a curator will review your submission and the summary statistics, and reporting and supporting metadata will be made available as soon as possible. For unpublished or pre-publication submissions, the summary statistics will be available within 24 hours or after the embargo date on our FTP file system. The study will then be fully searchable on our website after the next data release, which occurs every 2 weeks. And we'll make available the unreviewed metadata that you submitted, and that will be available to download also after the next data release. When your work is published and then gets a PubMed ID, we'll either pick it up in our literature search, or you can just let us know that it's out, and a curator will then review the submission and make the reviewed metadata available as soon as possible. So in summary, we accept summary statistics for any human GWAS with more than 100,000 SNPs. And this can be submitted either before or after publication of a journal article. Summary statistics must follow our standard format, with some updated requirements coming into effect at the end of March 2023. For step-by-step instructions, please see our website for more information. Stable identifiers are provided upon submission, and the pre-publication summary statistics are made available within a few days or when you give us permission to release them. Submissions for published papers will be reviewed by a curator and made available as soon as possible. So lastly, some questions that you might have about the new summary statistics format. So what if I've already started a submission and prepared my files in the old format? So in that case, you can submit the files in the old format until the end of March. If you can't complete the submission before then, but you don't want to start again, then please contact us and we'll see what we can do to help. What if I submitted summary statistics in the old format a while ago and they are under embargo or in a pre-published state? In that case, you don't need to do anything. Your data will be made available in the old format upon journal publication, and the metadata that we show on our website will be prepared from the information that you submitted. And finally, I made a submission in the old format, but I would like to update it to include the new fields. Can I do this? At the moment, unfortunately, our update process is a little bit too manual and we don't have capacity to update files unless there's really an error. But we do plan to improve this process in the future, so look out for updates.